All right, everyone. We're going to talk about genetics today. And I'm just going to throw out there that this lecture is super material dense. Um, and it changes a little bit from semester to semester. So I tried to mainly put what was common in both my semester and the semester afterwards into the slideshow because I'm not sure what will change. And what Dr. Himes normally does is she will send an edited PowerPoint out with a little X showing which slides will not be tested on. Um, so just a general disclaimer for all of that. And I'll try to go back through her slides also to see if there's anything that's drastically different, but most likely there won't be. So there's a general genetic continuum to determine whether something is completely genetic or completely environmental. So some things are completely genetic, right? There's a mutation on a gene, and it's just that one gene that causes this disease, like cystic fibrosis. And then some things are completely environmental. So, getting the flu is determined by whether you contract it or not. And it doesn't really have anything to do with your genes. Um, and then there's genotype versus phenotype. So genotype is represented down here, the picture on the left. So this is what do your genes say about who you will be? And then your phenotype is how is that manifested in reality? Because we know that, what happens isn't necessarily completely determined by your genes, right? Your environment has a say in it. So that is the difference between genotype and phenotype. Also, most of these pictures are taken out of Dr. Heim's slides. So some diseases are single gene disorders, like cystic fibrosis, and then others involve multiple genes and include environmental factors such as heart disease. Diploid versus haploid. So diploid are normal cells. Almost all cells are diploid. They contain, a copy of from dad and a copy from mom. So they have both two copies of each chromosome. And then haploid are only germ cells. So cells that become eggs and sperm are haploid, so that when they meet their other half, they can create a diploid cell. And then talking about some general definitions, a karyotype is up here in the top right. That's a complete visualization of all chromosomes in a person. So all 23 pairs of chromosomes are there in the karyotype. And then the chromosome is here. So here we see the chromosome in this picture. And then um, the gene is the specific part of the chromosome that codes for a certain trait. An allele is a specific version of a gene. So um, you can be homozygous, which means it's both like capital letter or both lowercase letter, or heterozygous, which means it's one and one for a pair of chromosomes like this. Um, and then there's hemizygous, which is there's only one copy of um, the chromosome. So, for example, males are hemizygous for the X chromosome because they only have one. So it's XY. And then the difference between penetrance versus expressivity. So penetrance is whether the gene shows or not. So here we see variable penetrance. Sometimes it shows, sometimes it doesn't. And that's kind of black and white. Does it penetrate or not? And then expressivity is, okay, we know whether it's penetrating or not, but to what degree and how? That's expressivity. And so you can have variable penetrance and expressivity where it's sometimes penetrating, sometimes not, and the gene is expressing itself to varying degrees. Epigenetics is how behaviors in our environment affect how a gene is expressed. So smoking is something that's from the environment, right? It's not determined by our genes, but it reduces the action of tumor suppressor genes, so you're more likely to get a tumor. So what you do can affect how your genes, whether they mutate or how they're expressed. And then de novo means that a trait popped up, mutated in a child that doesn't show in any either parent. So it's non-inherited. It's new. So novo is new. So it's a new mutation. And dominant versus recessive. So um, autosomal dominant manifests as a phenotype in those whose genotype is heterozygous for that allele. So here we see, the father is heterozygous for whatever orange gene this is, and he's affected by it. And all of his children that have that orange gene are affected by it as well. So they don't just carry it, they're affected by it. Um, and then autosomal recessive means that you need both of those in order for the child. Um, well, here this is confusing because now the one that's affecting is the purple one. But the child that has both purple ones is affected by whatever purple gene that is. And the child that has no purple ones is unaffected, and the child that has one purple one is a carrier. Okay, and then X-linked means that firstly, it'll only affect males because females have two X chromosomes. We talked about that earlier. Males are hemizygous for the X chromosomes, and females have both. And so when a female has the recessive trait on one of their chromosomes, the other one will overrule it because it's recessive. But for a male, they don't have another X chromosome to overrule it. And so it'll express itself. If the male has it, they will be affected by it. Okay, and then talking about benign tumors versus malignant tumors, pretty much just know what's on this list. So benign grows slowly. It's a well-defined capsule. It's not invasive. It's well differentiated. So lack of differentiation is um, almost always a worse sign than something that's differentiated. Low mitotic index, which means that there's few dividing cells and it doesn't metastasize. So it doesn't spread to distant organs. And a malignant tumor is typically considered to be worse. Um, can benign tumors kill somebody? Yes, of course. But malignant tumors grow rapidly. They're not as often encapsulated. They invade local structures and tissues. They're poorly differentiated. They have a high mitotic index, and they metastasize often through blood vessels and lymphatics. Okay, so now I'm going to talk about different cancer cell characteristics. So, anchorage dependent. Normal cells need to be anchored to another cell, but these cancer cells do not in order to multiply and live. Loss of contact inhibition. Normally cells, they're dividing, and then when they kind of hit a dead end, they stop dividing, but cancer cells don't. They just keep growing. And now I'll go through each one of these one by one. Um, so cancer and genomic instability, right? This gray one down here. The genes are more unstable in cancer cells, and so they're more likely to mutate. So that means that with age, mutations become more common because there's just more time and more probability and possibility that those mutations will occur. They induce angiogenesis, which means that they can secrete factors that'll create new blood vessels so that it can feed itself with blood. They can resist apoptosis. So normally when a cell's DNA is irreparably damaged, it'll self-destruct, but these avoid that self-destruction. Deregulating cellular energetics. So typically our cells use cellular respiration to create energy, which requires oxygen, but these can use purely glycolysis, which does not. Sustaining proliferative signaling. So normal cells proliferate in response to growth factors, but cancer cells can proliferate in the absence of those growth factors. Avoiding immune destruction. They find ways to escape detection so they can, for example, reduce the amount of antigens they present on the surface of their cells, among other things. They evade growth suppressors. So there's a tumor suppressor gene that makes sure that a cell does not multiply uncontrollably. But in cancer cells, a mutation can cause a loss, that gene to be lost. Um, Enabling replicative immortality. these cells can divide forever. They don't stop dividing in response to cellular stress or anything else. And so she talks about the story of this woman who had a cancer, and the woman has since passed away many decades ago, but her cancer cells are still dividing. So that's pretty incredible. Tumor promoting inflammation. Inflammation generally causes morphing of cells and put you at an increased risk for cancer. So one example is Barrett's esophagus, when your esophagus is chronically irritated by gastric acid. And so tumors can utilize this by releasing molecules that create inflammation and cause that continued mutation and multiplication. And then last but not least, they activate invasion and metastasis. So metastasis is when a cancer spreads to distant organs. So they can spread through blood, and the lymph system. And a primary cancer is where it originally started, and a second cancer is where it metastasized to.